Hello, everyone.
And folks are slowly joining in.
Yeah, exactly. We've got a longer list in waiting, so yeah.
Progress. Progress.
Good morning, good evening, good afternoon, wherever you are right now. We are so pleased to have welcomed you today on our very important talk on oncolytic virotherapy with Professor Chiocca, Professor Anthony Chiocca. So it's an honor to welcome him and to listen to him soon. So thank you very much for joining us today. We will be starting shortly. It's our speaker is going to join us in just a couple of moments. We just want to welcome you and make you aware that we are listening to you. I hope that you know about our initiatives and you also know about our YouTube channel with the ANS. So please to visit our archives, our video library at the ANS channel, and also subscribe to it to stay updated. We usually keep the recordings there so that you can go through them if you miss them, or if you want to go through them again and again, or you want to have to learn more. So most of the time, the we are currently providing webinars on various different topics that varies from basic topics for trainee levels, you can say, and also on innovation in gliomotherapy, brain mapping, and other things. We also have tumor boards. So at this time right now we are literally offering everyone a variety of topics that what we believe is in wrapping every probable aspect of neuro-oncology, neurosurgical oncology of our interest. So it's an honor to organize this innovation in gliomotherapy webinar. You can see that we have already conducted about I think 3 or 4 webinars previously on different topics. So you can go and see what we are headed to in terms of glioma therapy. So we are just going to start shortly once Professor Chiocca joins us. So we will start our webinar. So please feel free to share your questions as they arise in the chat box. So we can actually take them and discuss them at the end of the session. So I hope that you all know about the oncolytic virotherapy. It's a promising option. Still there's a lot of research going on, and everything is being under investigations that regarding this virotherapy and combination therapies that come through. So it's a basically quite pleiotropic, as we will discuss and you will see later. And if you haven't gone through, you can go through the relevant papers to have an idea to see what is it's all about, so that you can have a better idea of the question and how you want to, clarify your concept about this very innovative new treatment option. So Sir, can you hear us? Mr. Costas? Hi, sir.
Hey, good morning. I don't know what happened. That was weird. I,
Yeah, sir.
I think we had a tough time. Jennifer, there was a password it kept asking for me to put in, and I put the number, but I don't
Yeah,
know.
I'm not sure what might have happened. We are live. We do have attendees on.
Okay.
Dr. Chiocca should hopefully have the same
Yeah, he's joining
information.
on too. You're gonna put him on as a panelist?
That's right.
Okay.
Yes, I'm trying to find if he's in the in the attendee list, but he
It
hasn't
says
yet
he's in,
joined.
but it says he says I'm in, but it says on screen that I joined as a panelist.
That
Really? Let's
what
see.
is his name on here?
Anyo Kyoka?
No, I mean it could be different and
There is just one person, but it's not Dr. Antonio Chiocca. It's something Jacobo Antonio. I don't think that it's his account.
Okay, let's see. Let me see
what his name is.
Hi Nino, are you
And Antonio or maybe Nino, but I think that he will join from Antonio. Cannot find him.
Thank you everyone for your patience.
Yeah, sorry everybody. We've had a little bit of difficulty signing on here, but we're going to be joining momentarily.
Yes, exactly. I don't know how this arose, but it was a little weird. It's okay. Uh,
Are you looking through all the names? See if
Yeah, we just need to know what he's labeled as
I see. Uh,
because
yeah, it,
it's not self-explanatory.
yes, but if he has
let
sent
me
a
see if
screenshot,
I can call him. I'm going to go on mute. I'm going to call him real quick.
he has sent a message there. No. Sir, meanwhile, I believe that we can proceed with his brief introduction, or we should wait for him to join and proceed with his introduction then.
I just resent Dr. Chiocca another link to just join.
Yes, he has sent a message, but I cannot see the picture he has sent. He has attached. He said that this is my screen, but I cannot see the screenshot.
Right, he still needs to click on something else, so hopefully he'll join the next link I sent him.
I believe it, but we cannot I've also tried to find someone from Harvard, but there is no Harvard name as well. Maybe if he's using an official computer or something.
Nino Chiocca.
Okay,
Yeah.
Nino's getting on now as in the general session. So just try. It's Nino Chiocca.
We
Do you see
still
it
cannot
anywhere?
Sir, unfortunately, we cannot see him.
Okay, did you get on, Nino?
I just, it's the same screen. It says you've been invited as a panelist for Oncolytic Immunotherapies GBM. A topic, Oncolytic Immunotherapies GBM, select a session.
He just needs to join the link that I gave you, Dr. H, and then
No, I
he'll
know.
be
I
good.
gave him that link, Jennifer, and it says you've been invited as a panelist. There's nowhere for him to click. It's the email he sent us.
Jennifer, can you invite him directly through the link? Because I think there is an option in
That's the
the
link
by
that
the
I
host.
sent him. Mm-hmm.
Okay.
Yeah, for some reason he's not able to log on through that page. It doesn't have. He can't even say okay. It says check your email to find more information.
For me to come on this webinar. And if I recheck on that, it's the same link and I click on it again. Is it under, or the email, I'm either under Nino Chiocca or Chiocca@mgb.org. That's the other one that may be coming up. I don't know. Do you see me at all?
You don't see anybody on there for that has Chiocca on there, right? Maria?
No, sir, I cannot find anyone.
I just resent him his own panelist link. I
Oh,
just
he just
resent
sent you
it.
another one. Are you sending it to the Gmail, Jennifer, or to his MGB?
Get me registered as Gmail. Register me as Chiocca@bwh.harvard.edu.
Yeah.
I think the my Zoom, I don't have Zoom on Gmail and it's messing it up. I think.
Yeah, the Gmail is the issue. Okay, so here's what we're gonna do. I'm gonna.
My only problem is I have a hard stop at 8 because I gotta give another presentation.
I know. Well, I'd like to get you on here now in a minute, but geez. Okay, let's see. Okay, so this is MGB. I'm gonna give you his email.
Yeah, you can MGB or BWH. Try the old one.
I'll do that. Okay.
@bwh.harvard.edu.
Jennifer, I'm gonna reply to you and
Okay.
Maria with his bwh.harvard.edu email. Okay.
Sure.
Thank you, sir. I have his MGB number.
Well, I'm
email.
gonna do this one here. eachiocca@bwh.harvard.edu. Okay, here we go.
Chiocca.
Coming to you now.
Solid.
It may be the Gmail. We have issues with Gmail and where I work. They just do not like Gmail and they block us.
Yeah, I have to get that has to be changed back to my work.
Your work email. I think they had me like that too, Nino, at one point with the Gmail. Yeah.
Well,
Did you get it, Jennifer?
Yeah.
Yes. I've also sent a message.
Okay. Nino, try this.
I just resent him a link. He's got to see it.
Hopefully this works.
Yeah, he's there. Okay.
Okay, good. All right, we see you, I think, now. Yep. All right, you pulling him up?
Jennifer, can you please add him as a
Yes.
panelist?
I did, so he just has to.
There we go. Okay. We have liftoff here.
Thank you, everyone, for your patience.
Yeah, so sorry for the technological hiccup. It's not anyone's fault. It's part of it's a Gmail issue that I also have. But anyways, we're going to jump right into it here. We are so excited to have Dr. Chiocca come join us today. On behalf of the tumor section, I just want to welcome you all again for another exciting webinar. We put together a series of webinars that are quite educational and informative and cutting edge. And these will all be on our tumor section website, which has just been revamped, and also on NS. So Dr. Chiocca is going to be introduced by Dr. Maria Noor. But I also have had a long relationship with Nino, and he is just an outstanding neurosurgeon scientist, a real role model for all of us. And really excited. He has a new position as the new chair for Mass General Brigham, and just continues to propel our field forward. So Maria, do you want to provide a little more introduction for Dr. Chiocca?
Thank you very Thank you very much. It's a big honor for me. Exactly. So Professor Tony Chiocca is basically the executive director of the Center for Tumors of the Nervous System at NGB, and also the Harvey Cushing and Daniel Ponton Professor of Neurosurgery at Harvard Medical School. And he has got a lot of educational contributions, but his work, he's basically forerunner of oncolytic virotherapy, for which he also won the Golden Virus Award in 2023, which is basically one of the very several awards Professor Chiocca has ever been awarded. So basically, he's quite, like Professor Bustos had very rightly said, he's not only a neurosurgeon but a neuroscientist, and basically an innovator and someone who thinks a lot ahead and has a lot of vision. So it's an honor to have him today to talk about oncolytic immunotherapies in glioblastomas. And sir, thank you very much for joining us today. It's an honor to welcome you today, and the podium is all yours. Thank you very much. Sir, you are muted.
Go ahead and unmute. Yeah. There you go.
Okay. Can you hear me?
Now we can. Yes.
Great. Well, thank you very much for the introduction, and sorry for the technical issues. It is a pleasure to be here with you today. I'm going to discuss a project that we've been running for some time now related to oncolytic immunoactivation, and how we brought this from the lab, from the research lab to clinical trials. These are my disclosures. I'm going to skip them really fast, but we've been dealing with this fatal disease, fatal tumor called glioblastoma now for multiple decades. And we have tried several therapies for it. But even with new therapies, we have not really had much success. And even for therapies like immunotherapies that have been successful for other solid and liquid cancers, like the immune checkpoint inhibitors, for example, we have not been able to really make a dent in the survival of patients. One of the reasons why we think immunotherapies don't work is because this is a prototypical immunologically cold tumor or immunodeserted tumor. What does that mean? What that actually means is that when you take the tumor tissue, you see a rarity, very few tumor-fighting lymphocytes or T cells inside that tumor. The sine qua non for an immunotherapy to work is that you need to have a cytotoxic effector T cell in close proximity to a cancer cell, in fact, attached to a cancer cell through receptor ligand interaction, for the T cell to then inject into the cancer cell these enzymes called perforins and granzymes to kill off this cancer cell. And then the T cell can go on and kill other cancer cells. But in these tumors, we do not see that happen. We don't see T cells in the tumor itself. And therefore, even if you try to unleash T cells through immune checkpoint inhibitors, they just don't get into the tumor. So we've been trying to solve this, and this is my entire talk. If you have to get anything out of this talk, it's this one slide. What we're trying to do is we're trying to change this profoundly immunosuppressive microenvironment of the glioblastoma, which is full of these immunosuppressive myeloid cells with very few anti-tumor T cells. And by in situ intralesional administration of either an oncolytic virus or cytokine gene therapies or some type of anti-tumor gene protein, change that microenvironment into one that's much more propitious for immunoreactive T cells to come in and reducing the effect of immunosuppressive myeloid cells. So this is what we've been trying to do. So over the course of the last decade or so at the Brigham Dana-Farber, now Mass General, we have been performing several trials of modalities such as this, both in the newly diagnosed setting as well as in the recurrent GBM setting. And in terms of the recurrent GBM setting, the one trial I want to discuss is the one linked that says number 4 here, which is the which is the one of this oncolytic virus, and it's also here in the green. Let me make sure that I put my laser pointer here. If you see my laser pointer, great. So the way oncolytic viruses work is really through three direct mechanisms of action. The first one, it seems self-evident. It's a direct cytotoxic effect. These are pathogens that have been engineered to selectively infect and replicate in tumor cells. And therefore, the oncolytic virus will get into a tumor cell and cause a direct cytotoxic effect. But it's also recognized that that is actually probably a minor mechanism. It is needed to set off the rest of the immune system. But the second mechanism is activation of innate inflammatory responses. These are truly cytotoxic agents, and therefore you'll get a number of innate inflammatory responses, both cytokine-based as well as cell-based, to start coming on to try to remove the virus, but also remove tumor cells in the process. But lastly, I think the one part of the immune system that's really needed for these therapies to be durable and persistent is this getting the adaptive immune response to recognize not just virally infected cells, but also tumor antigens and tumor cells that have not been infected. And that is what the hope is here, that is this virus spreads in the tumor, causing more cytotoxicity, more inflammation, that you will get T cells start recognizing T cell receptors, tumor antigens, and start destroying that as well. So you could get a persistent vaccination response. And this is what we're trying to accomplish primarily, number 3. So there's been a lot of interest in oncolytic viruses with a number of different papers and clinical trials. There's the relatively famous recombinant poliovirus trial that was performed at Duke, published in New England Journal of Medicine, where they saw a tail of treatment responders. And then in 2021 and 2022, there were 2 trials of 2 other different oncolytic viruses. One was a herpes simplex virus type 1. The other one was an adenovirus, and this was for pediatric tumors. And then a couple of years ago in Japan, this particular herpes virus that's actually different from the one here, on the basis of this phase 2 trial, was approved by the Japanese FDA for GBM, for recurrent GBM. And then lastly, Fred Lang and Galera Zadeh published this combination trial, phase 2 trial of an oncolytic adenovirus plus pembrolizumab in recurrent glioblastoma. And therefore, there's been interest in these therapies from publications that have relatively high citation impact. So the oncolytic virus we have been working on for about 20 years was formerly known as RQ-Nestin 34.5V2. Now it's been renamed CAN3110 from the pharmaceutical company that's licensed it from my institution. But this is based on herpes simplex virus type 1. Herpes simplex virus type 1 is a common pathogen in humans. It infects neurons, infects neural cells, it infects epithelial cells. It causes the common oral labial sores. But it can also cause hemorrhagic encephalitis. And so therefore, a number of different HSV1s have been engineered for cancer therapy. And the gene that causes hemorrhagic encephalitis is called ICP34.5. And therefore, in all of the HSV1s that have been in clinical trials, but also there's one that's been actually approved by the FDA for treatment in melanoma, not GBM. All these viruses lack this gene because of toxicity, of worries and concerns about toxicity. The problem is that when you remove this gene from the virus does not replicate well, does not cause as much cytotoxicity. And therefore, we reasoned several years ago that we wanted expression of this gene, but we tried to restrict the expression of this gene that causes this neurotoxicity to the glioma by itself. And the way we did is we placed this gene under control of a promoter called Nestin, the Nestin promoter. And Nestin is highly expressed in glioblastoma, but in the adult brain, it is not expressed or expressed minimally. So the idea here is that if the virus infected a glioblastoma cell, it will still express this gene. It will cause a gliomitis, but it will not cause an encephalitis. And we do want inflammation in the glioma, so we want a gliomitis, without causing toxicity. And there's also safety features that allow us to shut off the virus. So these viruses remain very sensitive to antiherpetic drugs like valacyclovir and ganciclovir. So this is not for the faint of heart. This was done all academically through academic funds, primarily through the NIH. This shows my journey, the journey of my lab and my collaborators in getting this into humans. So in white, you see the original idea. And then we started first going to the biotech company approach, but that didn't work very well. And then we went totally through an academic approach and got funding by the NIH Alliance for Cancer Gene Therapy, as well as philanthropy, to get this basically to what's called an investigational new drug, an IND. We were able to send this to the FDA, and they finally approved the IND, and we treated our first patient with this virus in 2017. This slide is a little bit old, but as of the end of 2024, we've treated now a total of 57 patients. Now, to get it into clinical trials, we had to show through the IND that the data that we got into our lab was reproducible. And so we actually used a CRO, a contract research organization, to repeat our data. So these are mice with an intracranial tumor treated with our virus. And in blue, you can see the survival curve of these mice with a human glioma in their brain. These are athymic mice, compared to control mice here in red. So this was highly significant. One of the issues we encountered, though, was that when we injected this virus into the brains of mice, we did see toxicity. Even though the treatment was tolerated at certain doses, as you increase the dose of this virus, we saw toxicity. This is described in this one paper, but basically what you can see is that there's a tumor in this mouse brain right here. We inject the virus, and this by immunohistochemistry, you can see that the tumor is full of virus, which is great, but you can also see this lip of virally infected cells that go all into the ventricle. And it turns out that in mice, nestin, which drives expression of this neurovirulence gene, is highly expressed in the subventricular zone. And so we were getting basically a ventriculitis encephalitis in mice. So because of this, the FDA wanted to make sure that we did not inject the virus mistakenly into non-tumor bearing areas. And so we decided to use the ClearPoint MRI intervention apparatus in the intraoperative MRI so that we could see where the needle was when we injected this virus. And we used this smart flow cannula that has this tapered needle to do the injections. So this is a picture actually of a patient in the intraoperative MRI. My postdoc colored the tumor. This is a recurrent glioblastoma in purple. And you can see here the gadolinium enhancing area. She's in a prone position. You can actually see the stereotactic, the smart flow needle that's stereotactically being placed right into the tumor where we could do the injection. So we could actually see that the injection were in gadolinium enhancing area and not into brain, again, because of this concern and worry that you could cause an encephalitis. Now, the question is, we're using intraoperative MRI, should you just do a straight injection versus convection-enhanced delivery? We decided not to do convection-enhanced delivery for a number of technical reasons, and we would just do the injections of about 5 minutes in the intraoperative MRI scan. But because we're in the intraoperative MRI scan, we could just monitor for reflux. So this is a patient here. You can see actually this is a fairly long trajectory into this frontal basal area where there's the recurrent GBM. And what we could actually see is that as we did this, the bubble of fluid, which is shown here in dark, is being retained in the tumor, and we're not seeing much reflux out, even because of this tapered needle. And so that looked, and we did this repeatedly to show that we could actually get this fluid into the tumor without getting much in the way of reflux. So this is where we are with this trial. Again, these were patients that are recurrent high-grade gliomas. Most of them were glioblastomas. There were a few IDH mutant tumors, and there were a few recurrent anaplastic oligos, but it was really the minority of patients. So we first did what's called a dose escalation trial. We started out at a very low dose of virus, 10 to the 6 viruses, 1 million viruses. Again, the FDA wanted us to really go slow to make sure that we didn't get toxicity. And basically we went from 10 to the 6 low dose and increased it by half a log all the way to 10 to the 10th. Interestingly, we saw that this was highly well tolerated. We dosed 30 patients. We did not see any evidence of dose-limiting toxicity. We didn't see encephalitis, lethality like we had seen in the mice. And so the maximum administered dose was 10 to the 10th. So this was a single site tumor injection, one time point. And then we dose expanded. We got the FDA to allow us to go and inject 12 more patients. And we wanted to actually inject multiple tumor sites. Some of these tumors are large. We really thought that if we injected multiple tumor sites, we could get better biodistribution, not just of the virus, but also T cells and immune cells that would come in to fight off this viral infection in the tumor. And we did 12 patients dosed at this level. And we call this Arm A. This is our Arm A patients, which were published, as I'll show you in a second. And then we did another 9 patients in Arm B. We did these patients with pre-administration of cyclophosphamide to immunomodulate the patients. We thought that this would help us getting a better immune response against the tumor cells. And this is Arm B. And then we started a third trial, Arm C. And this is done multi-institutionally with 5 institutions throughout the United States through funding via this philanthropic foundation called Breakthrough Cancer. And the difference in this trial, instead of doing a single one-time point injection, we're doing up to 6 stereotactic injections over 4 months. So these patients are getting repeat dosing of the virus. And at the same time, we can get biopsies at each one of these time points that we can study via integrated multimodal genomic, epigenomic, metabolomic techniques to see what is happening in this tumor as we do these repeat dosing of the virus. So we can truly get a longitudinal picture of the evolution of the tumor as it's getting perturbed by this therapy. So the first Arm A was published at the end of 2023 in Nature, and you can see the results of this trial if you look up this publication. This paper is in preparation. Hopefully we should submit it soon. And this trial is halfway done. We've accrued to the first cohort, and we're about to start accruing to the second cohort. And again, this is being done both at the Mass General Brigham, but MIT is providing a lot of the scientific resources. Memorial Sloan Kettering, Hopkins, and MD Anderson are participating. So with Arm A, so I'm going to spend the rest of the time discussing this paper, what we found in Arm A, which really was 42 patients, 30 plus 12. And we didn't see any dose-limiting toxicity. This was, again, a phase 1 trial. We did see some serious adverse events that were possibly related to injection of the virus. Interestingly, they were both in young patients, and there really were seizures causing weakness. In this one patient, there was a little bit of a cerebral hematoma, which may have been caused by seizures. And this happened almost 2 weeks after injection of the virus. Both of these were controlled, but because they required hospitalization, they did become serious adverse events. Both of these patients were young, and they were IDH mutant gliomas and not glioblastomas. And then we had, everybody shows you the best responses. So this is an interesting patient. It was one of my patients who had a glioblastoma in the right frontal lobe that resected. And then he recurred, not just with the glioblastoma that we resected, but he also had a second tumor posteriorly as shown here. So we took him and just injected the second tumor, the new tumor. And this is the little bubble of fluid right at the brain tumor interface when we did the injection. And then over a period of about almost 12 months, you can see both of these tumors involuting. Even the one that was not injected went away. And we thought this is great. This is evidence of even an abscopal effect. You inject one tumor, and then perhaps you're getting an immune response against the second tumor as well. This is another patient. You'll hear a little bit more about her, but she had actually 3 surgeries. So this is her first tumor, right temporal GBM, 262 days before we enrolled in the trial. Take this tumor out. She undergoes standard care therapy, but then the tumor recurs. So 47 days before injection, we resect this tumor. But then, as you can see, it's starting to come back. So at this point, we decided to enroll in the trial. So you can actually see the needle here injecting into this recurrent area. And unfortunately, it looks like she recurs again. So at 30 days, 91 days after injection, you can see this area of enhancement. So we take her back to surgery and we suck this entire area. But this turned out to be primarily lots of lymphocytes, few tumor cells, but lots of immune cells. And she actually did really well for 2 years afterwards. Because of the immune cells in there, we did put her on Pembro for a while. But she had no recurrence. And as you'll hear in a second in this video, she actually lived a fairly full life. I'll tell you a little bit more about that. So this is a video of a couple of patients. The first patient will tell you a little bit about her story as she experienced this therapy. And then in the middle of this, it will stop, it will talk, and there'll be a narrative from the daughter of the second patient, the one I just showed you MRI scans. Unfortunately, the second patient that was tumor-free for 2 years ended up passing away, not because of her GBM, but she was involved in a fatal car crash. She was actually not the driver, but she was a passenger.
Nina, do you have some volume on there?
Beat the volume.
Yeah.
Sir, you might have to change the input into the video input instead of your own Zoom so that we may be able to hear it.
So I don't know. It sounds like the volume was an issue. But let me just go on here. So, basically, this trial was well tolerated. This was a phase 1 trial. In phase 1 trials, you really are looking for safety. But everybody in a phase 1 trial also is trying to get it whether there's a glimmer of efficacy. Are there any biomarkers of efficacy? So as part of this trial, we looked at a number of different demographics, serologic markers. And the one marker that really seemed to show that there was evidence of an effect was whether the patients were seropositive or antibodies to HSV1. Remember, this virus, this oncolytic virus, is an HSV1 based virus. About 2/3 of humans have antibodies to this virus. And as part of this trial, we were collecting serology antibodies before both pre and after injection. Now, previous trials of other oncolytic viruses, including the TVEC, the Imlygic that's been FDA approved for melanoma, never showed that serology, did not show that serology had anything to do with outcome. So we were actually surprised to see that our seropositive patients, and these are all GBMs, this does not include the IDH mutant patients or the oligos, that in our GBM patients that were seropositive, you could actually get a median overall survival of about over 14 months. Now remember, these are recurrent GBMs. Some of them were first, second, third, and fourth recurrence. Some of them were in thalamus. Some of them were multicentric. if you look at the median overall survival of a GBM that just gets of a recurrent GBM that just gets an injection, is not removed. It's been said historically it's about 7 months. And so if you believe those historical survivals, we basically doubled the historical survival with this injection. But we also had this internal control because the HSV seronegative patients, we had a survivorship for them, and it was actually 7 months, just like what the historical controls tell you. So you can see that we basically doubled the survivorship if you were seropositive compared to seronegative, and it's double compared to historical control. Well, this was interesting data, highly significant. And then I asked Alex Ling, who was my postdoc, a bioinformatician, as well as a biologist, to look more into this. And we said, well, maybe there's something different between this patient population versus this patient population. Maybe these are younger patients. Maybe they're, I don't know, maybe they have smaller tumors. Maybe they're MGMT methylated, blah, blah, blah. And so we did a Cox proportional hazard ratio. These are multivariate analysis. And we asked whether this seropositivity could be determined to be an independent prognostic marker of survivorship. And it turns out it could. So if you're seropositive, your hazard ratio of death was much, much less than it is if you were seronegative. This was highly statistically significant. And again, this was independent of everything else we could think of: tumor volume, dose, time from diagnosis, age, gender, NGMT, whether you use steroids, number of recurrences, or KPS. So again, this seemed to be an independent prognostic indicator of response. And then we asked the question, well, maybe these patients are different. Maybe they're just immunologically more fit. Maybe this has nothing to do with the injection of the virus. Maybe they will do this with injection of anything. So luckily, we were also collecting antibodies to another virus called HSV2. HSV2 is actually relatively different from HSV1. The genomic differences are about 30%. And some of the population is positive HSV2 as well. This causes genital sores. And when you collected the antibodies to this, what you can see in the same patient population, that there's actually no survivorship evidence whether you are seropositive or seronegative. So this turned out to be very specific to HSV1 and not to HSV2. We had further evidence that this was very specific to this particular virus, because at the same time, there was this paper that was published by Steve Francis, and Steve was from UCSF, and Steve was basically taking blood from all glioma patients, regardless of what therapy they were on, both recurrent and newly diagnosed, and checking whether there was any evidence of antibodies to any virus. And he was looking at VZV, CMV, EBV, as well as HSV1, had anything to do with survivorship. And what you can see is the HSV1 serology in this large cohort of, I think there was 10,000 patients, was not associated with GBM overall survival. So this seemed to be very, very specific to this particular virus, CAN310. It was not associated with just a general response of survivorship to antibodies to viruses. So the next question we asked is, can we get any additional analysis, informational, scientific analysis from these patients? And we really strived to get back tumors from these patients, either by resecting the tumor if we saw any evidence of more enhancement with time, so we thought if there was progression, or asking patients to donate their brain if they recurred and potentially died. And so we ended up having a pretty rich trove of about 32 paired samples. This is the tumor right before injection at time 0. And then sometime after injection, as short as 1 month after, all the way to 3.5 years out, we were able to collect these tumors. And now we have pre and post-injection tumors where we could do all sorts of interesting informatics. We could do just regular histology, but we could also do bulk RNA sequencing, cytokine analyses, sequencing for the T-cell receptor, and a number of different genomic analysis. We also collected plasma PBMCs from these patients as they underwent their treatment. And so based on this, there were 4 questions that we asked. The first question is, at the beginning, I told you that we inject this virus and we get an inflammatory response in these tumors. So the first question was, is there any evidence after injection that we do get more T cells in there, more immune cells into these GBMs? And so, as shown here. And so we do this by immunohistochemistry, and this is really work by Isaac Solomon, Jared Woods, Keith Ligon, and also Francesca Barone and Alex Ling. So you can do this visually. So this is one patient before injection, and immunohistochemically, we're looking at CD8 and CD4 cells. And after injection, visually, you can see, oh yeah, it seems like visually that there seems to be more CD8 and CD4 cells by immunohistochemistry. These are 3 different patients. When you look at where these CD8 and CD4 cells are, a lot of them are perivascular, as shown in all 3 of these patients. And that is thought to be because as T cells try to come into the tumor, they first come to the vasculature and they accumulate perivascularly before they start migrating through the tumor. There's also these large necrotic areas in GBM. Some of these were caused by the virus, and we saw a perinecrotic accumulation of each one of CD8, CD4, and CD20 cells around these necrotic areas of GBM. But because we had so many patients, we can actually do some quantification. Just instead of doing this visually, we could do this by quantifying it. So each one of these represents one patient before injection and after injection. And here we're quantifying the number of CD8 cells per cubic millimeter, or the number of CD4 cells per cubic millimeter, the number of B cells per cubic millimeter. And what you can see is that there was a statistically significant increase in these patients when looked in the aggregate on CD8s and CD4 cells, not as much for B cells. There were some patients where there were a lot of B cells, but there were also some where the B cells were not as much. So even though the average here seems to be different, the range here made it not statistically significant. Well, this is great. You can show that you inject the virus in the tumor, you get more T cells in there, which is how we started this project. I was telling you, we're trying to get more T cells in these tumors. Who cares? Is there any evidence that having more T cells in the tumor correlates with an outcome? And so the second study that we did, and this is thanks to Alex Ling, where we started correlating the number or the change in CD8 cells or CD4 cells in these tumors as a function of survivorship. So here I'm showing the change after injection in CD8s or CD4s, and here on the y-axis as a continuous variable. So we're not looking at long-term survivors versus short-term survivors. We're using this as a continuous variable, which is probably statistically much more significant of a result. And each one of these dots represents a patient, a paired sample, basically. And what you can see is that if you look at the seropositive patients, in other words, the patients that survived the longest, the ones whose median overall survival was about 14 months, you can see that this was statistically significant. The more CD8s and the more CD4 change there was in these tumors after injection of the virus, the better the survivorship of these patients. And again, these were the p-values. Well, that's interesting. Now we're starting to correlate a scientific correlate, the difference in CD8s and CD4 cells in a tumor with an outcome, survivorship. That's been hard to do in cancer biology, at least in human cancer biology. So we thought this was really exciting. But we then wanted to validate this even further. And so we did what I call the $200,000 experiment. We basically got genomic DNA from the paraffin blocks. So we isolated genomic DNA and then sent to this company called Adaptive Biotechnologies, and they sequenced the DNA in these blocks, and they can pull out the T cell receptor beta chains. So they're primers where they're basically sequencing just the T cell receptor. Each T cell has a particular T cell receptor. That's its signature, that's its zip code. And so a particular group of T cell receptors will basically say that this is a T cell clone. And so we wanted to see what was the change in these T cell clones that have this particular zip code, and does that correlate with survivorship? So similar to what I showed you before, if we see a change in these tumor T cell clones in these tumors, the bigger the change, the better the survivorship, and this was significant. We also can do more sophisticated analysis. You can look at these T cell receptors and see how diverse they are. Each T cell receptor represents a T cell clone. The more diverse the T cell clone, the better it's thought to be of a response, because it means that different T cell clones could attack different tumor antigen, for example. And so entropy is an indication of this T cell diversity. The more diverse it is, the better it is. And that's exactly what we saw. The more diverse these T cell clonotypes were, the better the survivorship of these patients. Now, the T cell clones that come to the tumor are coming from the periphery, right? They're coming from the blood. And so we asked the same question from blood, and we've got the same answer. The more PBMCs, the more of a change in PBMCs, T cell clonotypes in the blood, the better the survivorship. And again, clonality is an indication of diversity. Less clonal means more diverse. And so therefore, less clonality and more diverse T cells in the PBMCs, the better the survivorship. So this is great. So what I've shown you so far, we inject this virus, we can get more CD8s, more CD4 T cells in there. This correlates with outcome and correlates with diversity of these cells. So can we get any additional immunological information from these tumors? So one of the things we wanted to look at was whether the immune signatures in the tumors changed after as a indication of this virus, as a consequence of this viral injection. And so these are a little bit complicated graphs, but here we're looking by bulk RNA sequencing at what happens in transcripts in the tumors after injection of the virus. And basically, these are the tumor signatures we carried: anti-tumor cytokines, M1 signatures. So these are all immune signatures that we're carrying. And on the y-axis. On the x-axis, I put a p-value. So everything to the left of this red bar is a p-value less than 0.05. Everything to the right is a p-value of more than 0.05. So interestingly, in all the patients, what we saw is that there were several cytokines after injection that became significant. Several anti-tumor cytokines that were significant. However, in a lot of if you look at all the patients, there were some that were not. However, if you just looked at the seropositive patients, you can see that all of a sudden you get up to 13 different immune transcriptomic signatures becoming significant. So again, same type of thing. The seropositive patients show evidence of better survivorship, but also more immune signatures, especially when you look at all the patients, including the seronegative patients. So, so basically, I show you, we inject this virus, we see more T cells, we see more immune cells, we see a change in 2 immune signatures. I think for time purposes, I'm going to skip this because I see that we're running out of time, and just go to the final slide. So basically, how do we put all this together? So I think this is evidence of this virus working as a vaccine. It's actually as a booster vaccine. In other words, if you have been exposed to herpes simplex virus type 1, you probably do not have an active infection, but you do have T cells that are contracted, and you have this T cell memory population that knows that to respond if a virus infection occurs. And so we're giving to the patients that have pre-existing exposure to the virus a viral injection in their tumor. And now you have these T cells that just expand. They know that there's a virus there. And so as you inject the virus, these T cells rapidly expand, coming to the tumor and try to destroy tumor cells as well as virally infected tumor cells. And this gives us a better response. However, if you're seronegative, the immune system has to recognize, has to learn that there's a virus there. And by the time it has done that, the tumor takes off. So there's really not enough time for the patient to respond to this vaccine. So I wanted to thank you. Again, what I've showed you is that there's an inflammatory infiltrate that occurs in these tumors even after several months. We do not see evidence of neurotoxicity in the first 57 treated patients. The trial data showed that survival of patients treated with this virus associates with positive HSV1 serology. The seropositive patients' survival correlates with increases in T cells, T cell clonal types. I did not discuss this, but there were also specific T cell clonal types, more immune signatures, and I didn't show you this. There's also evidence that the T cells come in, they clear the virus from the tumor. So this takes a village to do, actually takes a city to do. So these are all the individuals and all the funding agencies that allowed us to go from the lab to the clinic. Thank you for your attention.
Awesome, Nino. I know you have a hard stop at 8, so want to thank.
That's okay. I have a few minutes.
You have a few minutes? Okay. Well, let's see what kind of questions come through. But one of the things that, the theme that comes out through your talk is that when we look at new therapies and we're trying to push the envelope, there's always challenges. And I think one of the points I'd like for you to kind of clarify is, we want to keep pushing a technology forward, but sometimes, we have to make a decision whether that technology is worth pushing forward, right? And I think, it's a difficult concept sometimes, right? Because you're vested in this technology and you've spent years doing it, and now you're seeing the fruits of it. But at the beginning, it was very difficult. Can you just briefly comment on that a little bit?
Yeah, no, that's a great point, and so basically, the median average from laboratory to clinic, first in human, it's about 10 years, even for pharmaceutical companies. For us, it took us 20 years, but we were doing it academically. And so if you decide on technology, you're married to that technology for at least 10 years. And naturally, in that 10-year period of time, other technologies come on board that may look better, that are improved, but you can't go back. Once you start this process, it's so expensive, so resource intensive that you really just have to push forward. And so, yeah, clearly with our virus, I think there are better viruses that we discover in our own lab as well as others, and there may be better ways of doing this. But again, once you start this, you kind of are committed. You are married and cannot get divorced with this technology for the next 10, 15 years. And it's something that it is hard to comprehend, but it's just the reality of it.
Maria?
Sir, I do have just one question. I won't take more time. Sir, sometimes we believe that if we combine virotherapy with other therapies in combination, especially the ICIs, the immune pointer inhibitors and CAR T cells, then we might have a better response, and especially since we have got some inherent antiviral response to the body. So, sir, what's your take on this?
Yeah, I think combination therapies is always in this, and it's always the way to do it. And as we think about how to proceed with this, we're interested in using combination therapies. The immune checkpoint inhibitor seems to be a natural one because they're FDA approved. And a couple of our patients, we did put on Pembro or Nivo after treatment, especially when we thought they were occurring. And that seemed to be well tolerated. We didn't have enough patients to see if it was statistically significant. But there's a potential of adding this to CAR T cells to get more CAR T cells into the tumor. There's a potential of adding this to vaccines, peptide vaccines, to get more response against different tumor antigens. So I think all these things will need to be done in combination.
And sir, what will be your how will you select patients for this particular therapy, and especially in regard to the immune status of that patient? Maybe that patient do not have a very good immune level, maybe an elderly patient, or there could be patient with autoimmune disorders. So how would you like to select those patients?
I think maybe you're referring to the seropositivity. I think we need to, we saw that the seropositive patients seem to do better. And we collected all those serology in a prospective fashion, but we analyzed the data. That was not something retrospectively. I think we need to do a validation study where we prospectively look at seropositive versus seronegative patients to see if this is truly a biomarker response. If it is, then we have to start thinking, how do we seroconvert the seropositive, the
Yes.
seronegative patient to seropositive patient? Because there's actually no vaccine available against HSV1. It's a natural pathogen, but unfortunately, there's no vaccine. And so, is there a way in which you can convert them? And, we've been thinking about that. I'm not totally sure I have an answer for it right now, but that's a good point.
Mhm.
All right, well, we're going to wrap it up here. I also want to just thank the WNS for their help with organizing these webinars and really getting the word out. We're really excited about continuing on with this. Thank you, Dr. Chiocca, for taking your time this morning, and sorry for the technological glitches. That's on us. We will fix that for next time. Have a great day.
Appreciate it. Thank you.
Thank you.
Thank you, sir.